নিত্যরূপ স্টার্ট করে দাও তুমি, লাইভ হয়ে গেছে কিন্তু।
আচ্ছা, a very good afternoon to our distinguished guests and to the members of the NIBMG fraternity. I welcome you all to our Institute Day celebrations. To start with, I request our NIBMG choir to initiate today's celebrations with an invocation song. I would now request our director, Professor Soumitra Das, to kindly deliver the opening remarks.
Sir, you are muted, sir. Sir, please unmute. Sir, first,
can you see the screen?
Yeah. Now it's coming.
Yeah. So, welcome to you all. Today's Institute Day celebration of the National Institute of Biomedical Genomics. I particularly thank our guest of honor and the chief guest, Dr. Shahid Jamil, and also the guest of honor, Dr. Sarah Tekman, and of course, Parthoda, our beloved Professor Parthopratim Majumdar, for establishing such a beautiful institute. Now, this institute was established in 2009 with the dream and vision of Professor Parthopratim Majumdar and the blessings from the then Secretary of the Department of Biotechnology, Dr. M.K. Bhan. And the vision was to enhance the knowledge on the human health and the disease through the practice of genomics, and to translate the knowledge using appropriate technologies for promotion of well-being and improvement of genetics-based healthcare. Remember, in 2009, the concept of personalized medicine, the concept of biomedical genomics was very new. And with that, the institute started, and now it's now 11th year, and we have 4 different verticals where we are actively participating and contributing into the research of the gene structure and functional genomics of cancer, the genetic determinants of infectious disease, the susceptibility and the pathogenesis, statistical and computational genomics, and the multiomics determinants of the complex disorders. And we have 14 faculties who are actually on the roll, and we also have senior scientists in the pool. And it's a relatively small institute, new institute, but actually we are addressing something very fundamental and something very, very useful for the translational medicines. We are actually looking at the genomes, 3 genomes and the environment. The 3 genomes are the nuclear genome, the mitochondrial genome, and the metagenome. And we are trying to understand how does this genomics knowledge can impact our health, try to improve the ability to diagnose the illness, to classify the patients to improve their treatment. As I was just saying, can we make it personalized treatment, reduce the public health burden, and enhance the national health index? Very recently, we have actually now more emphasizing on certain areas I would like to highlight. Otherwise, you have heard and seen all these slides many a times from me. So the single cell genomics, and actually Professor Shara Tekman will actually highlight on while she's going to talk about the human atlas. Each cell, we believe, their types, their states, their location, their transitions, and their lineage, all are important in terms of the genomic informations to really take an informed decisions about which way to go to address a particular problem in terms of treatment or in terms of predictions of a human health problem. Another thing that we are increasingly getting interested is to understand the human microbiome in the health and disease. Trillions of microorganisms, be a bacteria, virus, fungi, residing in the human body, and it helping in the performing important physiological functions. And this microbiome composition, the diversity of the commensal microbiome interacting with the host genome factors, leads to the development of a self-trigger in the chronic autoimmune diseases. Now at present, we are actually addressing using skin disease in human as an exemplar. We are also addressing the atopic dermatitis, psoriasis, diabetic foot ulcer, preterm birth, and the child growth retardations. And all of these things are actually done in collaborations with different hospitals and institutes across India. The third thing that we are actually interested to understand the microenvironment, particularly in the tumor heterogeneity. And we are trying to use the patient-derived 3D organoid models of solid tumors for preclinical studies. And can we really actually have, without, in absence of a proper animal model, can we really fall back onto this 3D organoid culture to have a real validations of the leads, important leads that we are actually obtaining from our sequencing results? The strength of this institute is actually collaborations. The institute is champion in coordinating multi-institutional programs. One such is the System Medicine Cluster, which we call SciMech, which is also called as a Kalyani Cluster. Now, there are actually 6 different institutes involved, and NIBMG is coordinating all these 6 institutes are ISA Kolkata, Bose Institute Kolkata, IICB Kolkata, ISI Kolkata, and Tata Medical Centers. And what we are collectively addressing is the oral and cervical cancer as an exemplar. We are also a part of the Grand Challenge program on the preterm birth, trying to identify the genomic and the epigenomic underpinning of the adverse birth outcome. We are also actively involved in the Genome India project, where we are trying to catalog human genomic variations with special reference to India. And we are also actually completed 1,000 SARS genome sequencing as a part of a consortium, and to obtain the countrywide landscape of the variations of the SARS-CoV-2 RNA genome sequencing. And all these things we are working, but there is the feeder line is the Kalyani cohort, which is a platform for research on complex trait and diseases, where we are monitoring 20,000 individuals drawn from villages in and around Kalyani, and try to understand, can we really get some informations from this cohort, which will help us in all these genomic studies. The flagship project of NIBMG is on the cervical cancer, which is also an exemplar for the artificial intelligence-based approach. NIBMG's AI-based non-invasive diagnostic approach will actually help to save the woman from succumbing from the disease by the early and accurate diagnosis of the cervical cancer and actually recommend the therapy regimens. We believe in collaboration, as I was referring. We are part of several international programs, International Cancer Genome Consortium to delineate the landscape of the gingival oral cancer genome, the ICGC project. We were part of this Genome Asia 100,000 genome to sequence and analyze 100,000 Asian individual genomes. We are also a part of the Human Cell Atlas, where single cell sequencing of oral cancers and the placenta will be actually also highlighted in Sarah Techman's talk late in the afternoon. We are also actively involved in a lot of outreach activities, as you know. Our focus is also in the Northeast. We routinely do Northeast region training workshops. We have organized in Assam, Tezpur, Mizoram, and Agartala. And each of these workshops are followed by advanced workshop where we actually identify some of those interested candidates, bring them here and train them more into advanced techniques of biomedical genomics. We have the National Genomics Core. Actually, it's 3-arm centers where the major center is at NIBMG, the other 2 centers in Allahabad and also in CDFB Hyderabad. The idea is to provide high throughput platform facilities and expertise for generations of genome scale data and provide facilities and expertise for big data storage, management, access, and analysis. We're just scattering over 80 institutes at present, and all this huge number of sequencing load is actually carried out with these national genomic centers. Another thing that we are helping the society is the national, we have a genetic service unit where we actually detect for the rare genetic disorders. And this essentially we are working with in tandem with the West Bengal government. And now we are trying to use it as a central facility with the MIT program to understand the genetic variations in the childhood days, or even in the pregnant lady, if we can really find out some genetic disorders and actually give them some solutions to that, and then have informed decisions. Very recently, we have several facilities been set up at the Institute. I'll just run through this quickly. One of them major was the zebrafish facility to confirm the functionality of the genes and regulatory elements underlying the human disease. Again, the leads that are obtained in the genomic studies, in the sequencing studies, can be confirmed and functionally validated using the zebrafish model. We also set up the proteomics facility at NIBMG, where we are started catering the neighboring institute as well. The BSL2 plus facility we have launched during the COVID time. Now we are aiming to make the BSL3 facility as well. As I mentioned, institute was actually coordinating the 1,000 genome sequencing effort, where we have actually covered 10 states, different zones within India, and completed in a record time. And we did show that how this, the A2A haplotype and with the D614G mutations overcasted other mutations. And we are now trying to understand the implications of these variations using different model system. And one more thing that we have very recently are coordinating is the Indian SARS-CoV-2 Genomics Consortium. It's an Indian consortium along with NCDC and 8 other institutes. Total 10 institutes are a part of this. And I'm sure Dr. Shahid Jamil is going to highlight in his talk when he talks about the COVID-19 in India. And this consortium, the idea is to continually monitor and track for the variants, whether some variants are actually emerging from India, or are we having the variants from other countries infecting our populations. We are actually trying to address sequencing of 5% of the positive samples daily to ascertain the current status. to establish the sentinel surveillance and early detection, and then trying to understand the implications of these genomic variants in particularly in terms of the disease severity and its susceptibility, or in the vaccine outcome. So as a small institute, we are growing, we are continually growing to newer heights. And what we intend in future is essentially trying to understand the cancer immunology and immunotherapy more, the tumor heterogeneity, as I mentioned, patient-derived 3D organoid culture, analysis of massive data sets using AI and machine learning, microbiome genomics integrations is another thing is in the cart, and finally the viral genomics, particularly the RNA virus like SARS and dengue, and what is their implications on the host. With this, I would like to conclude. Once again, welcome, and thank you all for participating in this event of this Institute Day celebration of National Institute of Biomedical Genomics. Thank you all. Welcome. Over.
Thank you, sir, for a wonderful introduction. I would now request the founder of NIBMG, Distinguished Professor Parthapratim Majumdar, to kindly take us down the memory lane and share his recollections and reflections.
Pattai, please unmute yourself. Pattai, you are muted. কি হলো বাস আনমিউট করতে পারছে না আনমিউট করে দাও
Are you able to see my screen?
Yes.
Okay. All right. So good afternoon. Very glad to be here. When the director of the institute, Professor Soumitra Das, asked me to talk about this, recollect the last 10 years, I was very hesitant. I still am very hesitant, primarily because memories are fading, and with the diminishing eyesight that I have, even reflections are very difficult to see. Be that as it may, it was very hard for me to say no to our director, and therefore I have chosen to accept this. And I will tell you how this institute was formed. And as Professor Das pointed out, the institute was initiated, at least the beginnings were in 2009. So that's when we started to think about this institute. The thought of this institute did not come de novo. It came through a telephone call at 7:30 in the morning from a person who is an icon, and he passed away last year, as many of you know. His name is Dr. M.K. Pan. He was at that time the secretary of the Department of Biotechnology. He had called to find out if I had any ideas about formation of new institutes that are specific on specific domains, very focused domains of science, because the Prime Minister of India at that time, Dr. Manmohan Singh, had announced that India was going to make some new focused institutes. 7:30 in the morning, did I have any ideas? I told him that I needed some time to think. So he said, in a couple of days, if you could give me a paragraph with your ideas. So my idea, I was kind of single track. I still have a single track mind. My mind is, scientific mind is all geared towards human genetics. So I proposed this, that we form a new Institute of Biomedical Genomics, essentially to talk about genomics in human health and disease. That one paragraph, after about 6 or 7 months, developed into a 100-page proposal, multiple rounds of discussion at various levels, including at levels of at international levels, eventually led to the formation of this institute. It was approved by the cabinet. It was formed as a not-for-profit society. It was to be it was to be an autonomous institution to be funded by the government of India through the Department of Biotechnology. So here we were at the end of 2009. I was still at the Indian Statistical Institute in 2010. I got a leave of absence from the Indian Statistical Institute, came over and started to think about formation of this institute or construction of this institute. Another person that I must recall at this stage is this person. His name is S. Sinha, and he was a comrade in arms, so to say, throughout the period, throughout my journey when this institute was being formed, and as long as I was in the chair of the director. He was the program officer, a senior program officer in the Department of Biotechnology in charge of our institute or establishment of our institute. So I'm very grateful to the icon of India, Professor M.K. Bhan, and to the senior program officer of Department of Biotechnology, Mr. S. Sinha. We started, initiating this institute, and our start was Spartan. I would not like to establish an institute if for the first 5 years I was only building buildings. So that wasn't the deal, and I started negotiation, and Dr. Bhan helped, Mr. Sinha helped, started negotiations with the government of West Bengal, and the government of West Bengal was very kind to provide us with space, a fair amount of space on the top floor of a building, which is a TB hospital, which is located not far from this place where we are now located. It's actually a 5-minute walk from this place, essentially beyond our boundary wall. So this is where we spent the first 6 years or so of our institute. We were very happy. We were a slightly smaller group of people, but we were very happy, and all of us were able to initiate our scientific activities from this particular place called the Netaji Subhash Sanitarium, which is essentially a TB hospital. I'm not going to talk about science at all because our director has given you an overview of the science that we do and that we plan to do. So this is where we started from, and then we started creating wonders. The kind of wonders that we created was first this is from the Google map, as you can imagine. The first wonder is this building that we are all in. This building from the top looks like a double helix, and this was a model that was given to us by an architect who eventually built the building. And this is a wonderful building, and there is no parallel of its kind anywhere in the world. Well, we do know of a little bit of double helical structures, both on Wellcome Trust and Sanger Institute and other places, but they are much smaller structures. This is a whole structure, and this is just absolutely stunning. If you see it as you're flying from Calcutta to Delhi, you overfly our campus, and you can actually see this double helical structure. We started from a campus like this, where essentially there was nothing. There was plantation. There was a banana plantation. We had to evict a few people, but be that as it may, it all ended well, and we started constructing. And this is, sort of middle of our construction efforts. And eventually, we came to a building. And as we entered the building, we are in awe, precisely because we encounter another double helical structure. And this double helical structure was created by somebody who is not very well known, a sculptor who is not very well known, but works in the city of Calcutta with the National Council for Science Museums. So this was just absolutely stunning. As you enter the building, you not only see the building from the top if you're flying in from Delhi to Calcutta, which we hope that Dr. Shahid Jamil and Dr. Sarah Teichmann will be able to do shortly after the pandemic gives way for travel. And then as you enter the building, you see this stunning sculpture, and this is so we were creating wonders, and that's one of the visions all of us collectively had. We also create wonders in terms of our students who can sit on the footsteps of Watson and Crick. And as you enter the building, if you look to your right, you will see Watson and Crick, and you will see students sitting on their footsteps or below their footsteps and trying to understand genetics, which is again, something that's, the atmosphere is just fantastic. And we have created wonders even there. This was made possible by the visit of a Nobel laureate, Harold Varmus, who visited us in 2018 and actually inaugurated our Human Genome Hall. The Human Genome Hall is something that's very unique. No other institution, at least in India, has a museum that's devoted to science, and this is devoted to genomic science. And this was inaugurated by Harold Varmus. And here he is again wondering at the various kinds of exhibits that there are in the Human Genome Hall. As we not only create wonders, we mesmerize people. We mesmerize our students, we mesmerize our visitors, and the biggest mesmerizer of them all was M.K. Bhan, who actually established this institute. We are very grateful to him. Our teachers mesmerize, and look at this. I mean, this is look at their eyes. Both in on the left is Shobhik Mukherjee, who is a faculty member of the faculty of the institute. And if you look at the eyes of the students, they are all mesmerized. So our teachers mesmerize not only in the labs, but also in the classrooms, and we mesmerize our visitors. This is Terry Speed, fellow of the Royal Society, visiting us, and he is, of course, being mesmerized by Aurindam Moitro, giving him a tour of our labs. So we mesmerize our students and we mesmerize our visitors. I'm using the word mesmerize in a very academic sense, and we mesmerize them with our science. We love to march forward together, and this march forward is again not just in science, but also in terms of our being together socially. And this is our long march, and we love to march forward together. We hope that this kind of march will continue as long as we exist. And it is this cohesion among students, amongst faculty members, that builds an institute. And what I would like to reflect, let's say 10 years from now, is to see longer marches together, going forward, moving forward our science and our cohesion. We derive immense happiness when our students, after having worked for 5 years in the institute, are able to submit their PhD thesis. And here are the first 2 students that came out of the institute, and we have many more now. So we derive immense pleasure when our students are able to come out submit their PhD thesis, defend their PhD thesis, and get Dr. Dot imprinted in front of their names. This really gives us immense happiness and pride. We engaged locally. One of the things that we thought is that the institute is located about 50 kilometers out of the city of Calcutta in a sort of remote area, in a peri-urban area, and we can't be like an island, esoteric island in the middle of all of what we see around, which is not very rich neighborhood, which is a sort of middle class and poor neighborhood. So we need to engage with them, otherwise they will look at us as foreigners, and we will never be able to engage, will never be able to enmesh ourselves with the local society. So we started a program which our director alluded to, which is called the Kalyani Cohort Study, where our field investigators, we ourselves, our students, they go and meet with them from time to time, collect data, collect blood samples, and this is the longitudinal study of 40,000 individuals that we have, captive individuals whom we have been following up for the last 10 years or so. I'm not going to discuss the science of what we have derived by studying them, but this is our way of improving our own science, but also at the same time engaging with the local community. We love our trees and more. Every visitor who comes plants trees, and here are trees being planted by many of our visitors who come, and we like to keep our campus absolutely green, and we like to retain the trees that are there on campus and plant more trees. We love visitors, and we have had stellar visitors, and you have seen this slide of mine many times, but at the same time, this year we have new students. So let me point out, I don't know if you can see my pointer. This is Mary Claire King, who discovered BRCA1, the breast cancer gene 1. And this is Mike Stratton, director of the Wellcome Sanger Institute, who is the discoverer of BRCA2. We've had stellar people. Eric Green is the director of the National Human Genome Research Institute of the U.S. National Institutes of Health. Arvind Chakraborty is the director of the New York Genome Center. Steve O'Brien spent his lifetime at the National Cancer Institute in the U.S. National Institutes of Health. Right now, director of a genome center in St. Petersburg. Professor Obaid Siddiqui passed away, the father of molecular biology in India. And Lou is the president of the what's the cell line? I forget. Establishment which houses all of the cell lines. It's in the U.S. Peter Campbell is with Wellcome Sanger Institute. Sir Walter Bodmer is one of the founding fathers of population genetics. So we've had many other people. This slide will not accommodate them all, and we have had the Nobel laureate Venki Ramakrishnan visit us and speak to our students, speak to our faculty, and so on. We are picturesque. We are picturesque in more ways than one, like I explained to you or presented to you. We are a bunch of awesome people. We mesmerize. We make our environment green. We engage locally, and we are also picturesque because we take pictures, and we have wonderful flowers on our campus. And we, like I said, that we love our visitors, and we welcome today's chief guest and guest of honor to come and visit us as soon as the coronavirus will permit. Thank you very much for your attention.
Thank you, sir. It's a privilege to hear about those experiences directly from you and, of course, to be a part of this institute. We move on to the two key lectures of this afternoon now. First, we have the Institute Day lecture, which will be delivered by our chief guest, Professor Shahid Jamil. I would request Dr. Shobik Mukherjee to kindly introduce Professor Shahid Jamil.
Thank you, Diptarup. It's my pleasure to introduce Professor Shahid Jamil. He is an eminent scientist and an Indian virologist. He is currently the director of the Trivedi School of Biosciences at Ashoka University. And before that, he was the chief executive officer of the Wellcome Trust this thing, and Wellcome Trust DBT India Alliance. And he is known for his research in hepatitis E virus. And he is a fellow of all the three National Academy of Sciences. And in the year 2000, CSIR has awarded him Shanti Sharup Bhatnagar Prize for Science and Technology, one of the highest Indian science awards. Thank you. Thank you, sir, for giving this Institute Day lecture. Over to you.
Thank you very much, Shobik, and thank you, Soumitra, Partha, and the NIBMG family for inviting me to give the Institute Day lecture. I'm also very pleased to be here at the same time as Sarah. We shared a course together a couple of years ago at the Wellcome Trust. So let me start sharing my slides and get on with it. Okay, so I decided to speak about COVID in India and give a fairly general talk, but of course I realized that I'm speaking to some very high quality genome sequencing people. So I will also step into their territory and talk about the COVID genome variation, the SARS coronavirus genome variation, and the variants, and everything that's going on in India on a daily basis. Every time one mutation pops up, it becomes front page news. So the outline of my lecture today is that I'll first speak about the pandemic in India. What sort of numbers are we looking at, both confirmed cases and possible cases, and where do those come from? How many infections do we have in reality? there is much speculation in the media these days that we may be looking at a second wave. So are we looking at a second wave? What is the evidence? And then I'll move over to really my comfort zone, which is biology of viruses. I'll speak a little bit about the SARS coronavirus 2 biology, and that will flow into genomic diversity, clades and lineages. I'll speak just very briefly about the kinds of vaccines that are being made and the vaccines that are available to us in India. And finally, try and address whether vaccines would really work against variant viruses. All right, so as of yesterday evening, the world had seen over 111 million cases, confirmed cases of COVID, with almost 2.5 million deaths. This morning, the most developed country in the world, which is the United States, crossed 500,000 deaths due to COVID. And as you can see, they have almost 28 million confirmed cases. Well, India at this time numerically ranks at the second position in terms of number of confirmed cases, and at fourth position in terms of number of deaths due to COVID. If you look at the bottom right-hand corner, there's a yellow graph which shows you daily cases. And you will see that the daily cases across the world, that's the world average, is going down and has been going down now for a few weeks. But then you're also aware that there are places in the world where cases have risen. But thankfully, in UK, cases are now going down, and US is also showing a downward trend. Looking at numbers more closely, and I'm more interested in numbers from India. India has about 11 million cases, 156,000 deaths. So the case fatality ratio in India is 1.4%. And a recovery rate of 97.2%. The case fatality rate in India has been touted as one of the lowest in the world at 1.42%. This is about half the global average. The global average is roughly 2.5%. However, it would be wrong to say that India alone has a low case fatality ratio. If you look across Southeast Asia, you'll find the number anywhere between 1.2% to 2%. If you look in our immediate neighborhood, if you look at Pakistan, Nepal, Bangladesh, Sri Lanka, Myanmar, all of them, except for Pakistan, which is slightly higher than us, every other country in South Asia has a case fatality ratio that's lower than ours. And of course, numbers of infections are also much lower in those countries. So one question, obviously, that has been debated very much in the Indian media, as well as in media worldwide, as to why India has so few deaths. There's been much speculation that India is not recording its deaths properly, and without going into all that, even in the best of times, only about 20% of deaths in India are actually recorded. So people who are dying in hospitals, of course, you can't miss them. But those who are dying maybe at home of unknown causes, possibly COVID, that will not be recorded. Nevertheless, there could also be biological reasons for why the fatality rate is lower in our part of the world, and various reasons have been attributed to it. people have talked about BCG vaccination, and there's really little evidence for that. But the general thinking is that in our part of the world, when you live in environments where you are uh, where you are subjected to many other infectious diseases, uh, your innate immunity remains high, and if your innate immunity is able to control the virus in your nose and throat and doesn't allow it to get into your lungs, then possibly it doesn't cause more severe disease and less mortality. Very recently, a paper published by Partha and his group have also presented another hypothesis, and I will deal with that just a little later. So that's really the scenario from our part of the world. If you look at this graph and see how India did in time compared to other countries, so initially you see US and Europe were up. As India started going up around August and September, US and UK were pulling about half the rate of India. And then India started coming down, and Europe and US have continued to surge. So that is the situation right now. India is pulling roughly 13 or 14,000 cases on a daily basis now, compared to, 200,000 plus cases in the US. All right. But then there is apprehension that the cases in India are rising, and there is speculation whether this might amount to a second wave. So if you look at the top graph, and this is from this morning's Indian Express, those of you who are interested may go and read this article. It's a nice summary of what's happening. Since February, the daily case load in India has sort of gone up and down. But over the past maybe 1 week, the number of cases have continued to go up. And we're pulling roughly 14 to 15,000 cases on a daily basis now. But much of this rise is really coming from two states, essentially, Maharashtra and Kerala. Maharashtra continues to surge. And if you look at the total figure, Maharashtra's contribution to India's total COVID caseload is about 20%. So one state has been really the most affected. Kerala has really been an enigma because Kerala did so well in the early part of the outbreak. The daily caseload in Kerala actually went down to zero when the rest of India was surging. And then they started picking up. So it's possible that, since they did so well early on and there were very few exposed people, when they started getting an influx of people from other states, Kerala started picking it up. And of course, you can't rule out COVID fatigue, you can't rule out complacency and all of that. So India is rising again, and what I show you in the blue line here is the active caseload on a daily basis. So you see that since the first of this month, the caseload has been coming down, and it's been coming down continuously since mid-September. And this is really the enigma that while most other countries have sort of gone up and down in time, India has had such a prolonged decline over a few months, but is now picking up. And this, some people are worried, could be our second wave. The bottom figure shows you that for the first time, the dark line is higher than the lighter line. The dark line is new cases. The lighter line is the daily recovery rate. So essentially, the daily recovery rate has now fallen below the daily caseload. And that is currently worrying many policymakers. So the question really is, how many infections are there really in India? We're talking about 11 million cases in India, but how many cases are there really? On 22nd of September, ICMR released its second sero survey, which showed that 6.6% people across 70 different districts in India had antibodies. 6.6% translates into about 90 million people. At that time, the confirmed case load was 5.6 million. So if you do very simple arithmetic, you come up with a factor of 16. So if you multiply confirmed cases by a factor of 16, you arrive at the figure that was shown by antibodies. The third ICMR sero survey came out a few weeks back, and it was really surprising because the way India's caseload was going down, many people were thinking that there would be 50-60% seropositivity. The third ICMR sero survey showed that in adults, it was 21.4% seropositives. In 10 to 17-year-old children, it was 25.3%. So firstly, we were showing that children also get infected and children also transmit, but children don't have much symptomatic infection, just like everywhere else in the world. But we were not even close to what could actually amount to a reduction in the cases simply by prior exposure. And you will realize that I'm consciously avoiding the term herd immunity because it really has been a very, very misused term in the current pandemic, at least in our country. But then if you look at the finer points of this ICMR sero survey, you realize that in the third sero survey, most of the major urban centers were excluded. So Delhi was not there, Mumbai was not there, Pune was not there. I believe Chennai and possibly Bangalore were there, but beyond that, I don't think any of the big cities were there. But then when you look at data that's shown in this graphic on the right, you find that as early as July, Mumbai, the slum areas of Mumbai had reached almost 60%, the non-slum areas almost 20%. Delhi's sero survey, the third Delhi sero survey is not shown here. It showed 57% on average across different areas of Delhi. So possibly the number could have been higher in the third ICMR sero survey if some of the cities were also included. So one of the things that this is telling us is that possibly the outbreak has moved away from urban centers into villages where there really isn't enough testing and we don't really know what is happening. It's also, but the downward trend of the curve suggests that the outbreak is not really moving as much in the urban centers where it moves very quickly, but is moving more in rural areas where as it is, there is very poor testing and therefore you're not really picking up everything that you should be picking up. So yesterday, if you look at confirmed cases, 11 million, then just simply multiplying by that factor of 16, you get roughly 176 million people who possibly have been infected. But there is some more data, and this data is actually not publicized very much. There's this pathology testing lab called ThyroCare, which back in July released results of 60,000 antibody tests across 600 pin codes in India. And they found that the average positivity, mind you, in July was 15%, and the range was 0.7 to 47%. This is July. The second ICMR sero survey, which was around that time, is showing sero prevalence of only 6.6%. So it's more than twice. This is what ThyroCare was showing. And the ThyroCare sample size was bigger. ICMR sero survey was done on 28,000 people. ThyroCare did it on 60,000. And so that's the number. So 15% of India in July was 200 million people. They went on and did another in August. And this time 200,000 tests showed 24% positivity. So mind you, if you believe in that, then in August, about 330, 350 million people in India had already been exposed to the virus. And if you go from that time till now, it would possibly be safe to believe that somewhere in the range of 400 to 500 million people in India would have been infected by now. So keep that number in mind. All right. So let's now switch to the biology and genomic diversity. Well, this you're all very familiar with. I just wanted to very briefly highlight the point that the nucleotide identity of this new virus was highest with the bat coronavirus that was isolated in eastern China in 2018. And at that time, there were predictions that something like this might break out, but of course, nobody paid attention. Has only 80% identity with SARS-CoV-1 and much less with the MERS virus and the common cold coronaviruses. As you are aware, coronaviruses are RNA viruses, and they are single-stranded RNA viruses, which have the largest genome of any RNA virus. Another distinguishing feature of this is that unlike all other RNA viruses, coronaviruses code for an exonuclease that is used for error repair. It has an error repair function. So as a result of this, the mutation rate of coronaviruses is the lowest of all RNA viruses. Now, the virus is decorated on, it's an enveloped virus. It's decorated on the surface by three proteins, envelope, membrane, and the most prominent is the spike protein that you see here. And it is the spike protein that comes together as a trimer and binds to the cellular receptor, which is the ACE2 receptor. So this binding of the spike protein to the ACE2 receptor is something that is required for the virus to enter cells. And then, of course, there is a protease cleavage, and I'll come to that in a minute, which enhances the ability of the viral and cellular membranes to fuse together so that the viral core, the genetic material, can enter cells. And naturally, all vaccine approaches, therefore, are developed against preventing the binding of the virus to its receptor. So if you have antibodies that bind to the spike protein and prevent binding of the spike protein to the cellular receptor, then you can prevent entry of the virus. That's really the common theme across all the vaccines that are being built, all the vaccines that have been approved. So 30 kilobase genome, the N-terminal, or rather the 5' two-thirds of the genome codes for non-structural proteins, These are proteins that have enzymatic functions, the most prominent of this being the RNA-dependent RNA polymerase or the replicase. But you see, you also have an exonuclease shown over here, and many other proteins. A protease is there, a pepin-like protease is there to mature the virion, and so on, much like most RNA viruses. The 3' 2/3 of the genome codes for the structural proteins shown in purple. So you have the spike, envelope, membrane, and nucleocapsid, and some regulatory proteins, the ones shown in green. ORF3A, for example, is a protein that makes an ion channel, which is important for viral infection and viral pathogenesis, essentially. So coronaviruses make anywhere from 24 to 27 proteins, and these are really the prominent proteins. I mean, the one we will focus on for today's discussion is the spike protein. A bit about viral replication, and this is a busy slide, so let me walk you through it. The first step is binding of the virus to cell surface receptors. And the receptor, as you know, is the protein called ACE2. When the virus binds to the receptor, it is, the spike protein is cleaved at the surface by the protease TMP tempress. And following that, there is a fusion of the viral membrane and the cellular membrane, due to which the virus enters into an endosome. The core of the virus enters into the endosome. And following acidification of the endosome, the nucleocapsid is released. And therefore, drugs that prevent this acidification, drugs like chloroquine, hydroxychloroquine, and azithromycin have shown some effect, although large clinical trials have really shown no benefit from these drugs. Once the viral RNA is released, this being a positive stranded RNA virus, the RNA is translated into the non-structural proteins first, and these non-structural proteins then convert the plus stranded RNA into minus strand RNA, and the minus strand back to plus strand. That's the amplification step of the genome. And drugs that inhibit the RNA-dependent RNA polymerase, which is the enzyme required for this process, drugs like remdesivir, favipiravir, and ribavirin have also shown variable effects on the virus. Unfortunately, these drugs have been used very late in infection, and that's when they have not shown much effect. And you'll realize that for a replication inhibitor, it has to be used early before the virus is replicated to sufficient numbers. Anyway, after this, when the negative strand is converted into plus strand, it makes a genomic plus strand, but it also makes subgenomic RNAs. And it is these subgenomic RNAs that make the structural and the regulatory proteins that allow the virus to assemble into new particles, and these new particles are released. Oseltamivir is another drug which is essentially a flu drug, which interferes with exocytosis. So again, it has shown some variable effects. So that's really the life cycle of the virus. Some other drugs that have been used with success are steroids, for example, or therapeutic monoclonal antibodies, but these don't interfere with the viral life cycle. Steroids prevent inflammation, and monoclonal antibodies neutralize the extracellular virus, but do nothing to virus that has already entered cells. Okay, so if we look at genomic diversity, the highest amount of genomic diversity you see are in the structural genes, and naturally so, because these are the genes that are under the most amount of selection pressure, and especially the spike protein, because that's the target for immune response. That's also something that binds to the receptor, so binding has to be optimized. But the evolution rate for coronaviruses, and this is calculated for the COV1 virus, turns out to be fairly low. So 2 to 3 nucleotides per month is the mutation rate. And if you compare this to something like influenza, which is at least 1,000 times higher than this. But the fact is that since there is so much infection, and at least 100 million people have been infected, there's a lot of virus floating around, and therefore a lot of opportunity for mutations to happen. As the virus came out of Wuhan, it started adapting, it started changing for better infection, and developed various clades. And very quickly, the original clade, which is shown here, which is called 19A, 19B, diversified into into the 20 arm of this phylogenetic tree, and the virus has continued to diversify. The proportion of each of these clades contributing at any given time is shown in the illustration below here. And what is also shown here are the UK variant shown here, the South African variant, I believe, is this one right here. Yes, and the Brazilian variant over here. Another way of representing this on a radial map, the ones closer to the periphery are the ones that are the most recently developed clades, and you will see how distinct this particular, the UK variant clade is, and how late it has developed in the history of COVID, and so is the Brazil variant, and so is the South African variant. How about India? Well, India has, as you know, 11 million cases. In the GISAID database, there are 5,261 viral sequences, which amounts to 0.05% of confirmed cases. So far, 81 UK variants have been detected in India. And this is again already in the public database. So far, there is no report of the UK lineage virus coming from anyone who has not had a history of travel. But it's possible that that picture may change in future. But this is the factual situation now. The other reason I wanted to show you this is because for a long time, there's been this thinking that the virus that circulates in India is a weak virus, and that's why we have such low levels of mortality. Well, if you look at the virus in India, although only about 50 odd viruses are present on this tree, the virus circulating in India is present across the entire phylogeny of SARS-CoV-2. So that hypothesis is really not the correct one. And At this time, these are the mutations that have been seen in India. The N501Y, which is a defining mutation in the UK lineage, has been found in, and this I'm not talking about the UK variant lineage, I'm talking about from non-travelers. So this is the situation right now. The mutation that has created a lot of stir in the past few days is this mutation E484Q, which has been detected in 4 people in Maharashtra, sequenced at the BJ Medical College in Pune. And because of this, certain newspapers and certain experts have started saying that, it is this variant that is responsible for the increased caseload in Maharashtra. While there is no clinical correlation that has been drawn so far, and it's been seen only so far in 4 patients. So it would be wrong to derive that conclusion. Soumitra told you about INSACOG, and I'll come to INSACOG in a minute. All right. So another paper that was published very recently, which summarized the status of all the sequences from India, and this is a paper from CCMB. It showed, of course, a timeline for the emergence of clades in India across different states and across the country as a whole. But I want to focus here and look at the frequency of mutations across the genome that has been found in India. And you find a lot of ORF1A and ORF1B mutations, which are likely to have some effects on replication rates, but also significant mutations in the spike. And in the spike, if you break it down, there are some characteristic mutations in the N-terminal domain, and a number of mutations are focused around the receptor binding domain, which is really natural because the receptor binding domain is the one that adapts to bind to the receptor. This is also the site where neutralizing antibodies will bind. Now, the mutation that came about very early in the pandemic is the D614G mutation, which arose sometime in January and has really taken over the world. And if you see the bottom illustration here, about 99% of the virus circulating now contains a glycine at 614 position instead of an aspartate at that position. Work has shown that the G614 virus replicates better than its D counterpart, and this has been tested in cell culture. And in cell culture, you see that if you infect with equal amounts of both virus in culture, the output is that the G virus, there's more G virus than the D virus. The same thing has been tested in a human airway model, which is more similar to the bronchial airway tissue. And again, the same result that the G strain replicated better than the D. And the G really outcompetes the D when you give equal amounts of it. The G virus is also fitter than the D virus, and these experiments have been done in a hamster model where both viruses, hamsters have been inoculated with both viruses, and the nasal washes show higher concentrations, higher titers of the G virus compared to the D virus. But thankfully, neutralizing antibodies from people who have recovered from infection neutralize both equally well. So far, so good. And here is the paper that Partha and his colleagues published from NIBMG very, very recently, where they have put forward a very interesting hypothesis as to why the G virus is so much better at infection. And their hypothesis is that G at this site at 614 creates an additional elastase site in addition to the cathepsin sites that surround it. Because of this, the virus has the S1, S2 parts are cleaved far more efficiently when you have a G at that position. And they went on to correlate this to the presence of alpha-1 antitrypsin deficiency in people across different parts of the world, because as you know, alpha-1 antitrypsin is an inhibitor of elastase. So when you look at these frequencies, the frequency of alpha-1 antitrypsin deficiency is much lower in East Asian populations compared to European and North American populations. And they also show rates across countries in the different regions. So really the model, the hypothesis they have, which they've presented in the paper, is that in individuals with normal levels of alpha-1 antitrypsin, the elastase in the lung, the neutrophil elastase in the lung is inhibited by alpha-1 antitrypsin and does not get an opportunity to cut the S1, S2 junction in the infecting virus. However, in individuals with alpha-1 antitrypsin deficiency, there's much less of the inhibitor, making the elastase much more available and active to cleave the virus that has G at that position instead of D at that position. And this leads to enhanced viral entry. This leads to, of course, more inflammation and tissue damage. I was trying to find the serum elastase, serum alpha-1 antitrypsin levels of in populations across these three these three populations. I could not find that, but maybe I'll seek help from Partha and his colleagues to look at that. So interesting hypothesis, which explains why this particular mutation has essentially taken over the world, and why it may be so, maybe less lethal to people in East Asia. So just a word about the lineages, and there's also been a lot of confusion about variants and lineages, variants, mutations, lineages, clades, and these have really been used interchangeably. So lineage is essentially a number of mutations coming together, and the most popular one going around these days is B117, which is also called the UK variant, which has now been detected in 83 countries. Sequences are available from 64 countries, and it has 23 mutations, 17 non-synonymous mutations, and out of those 17, 8 are in the spike gene. These are just maps showing timelines as well as whether the mutation, whether this particular lineage has only been seen in imported cases or is also locally transmitting. And for us in India, at least so far, we don't find any local transmission happening yet. The B1351, which is also called the South African lineage, actually brings together two receptor binding domain mutations in the same virus, which is the 501 mutation and the 484 mutation. And that seems to be a cause for concern at this time. And there are other lineages. P1, for example, is the Brazilian lineage. And there are two other lineages, the 525, which was first seen in California, but it has spread across other places, and another lineage 23.1. Okay, so what about vaccines? The types of vaccines in development, well, you have the whole virus vaccines, both inactivated and live attenuated. A couple of inactivated vaccines have now reached clinical testing, and at least the Indian one has and two Chinese vaccines have also been approved for use. The genetic vaccines are the ones that have really captured everyone's imagination, especially the RNA vaccines, which have become, which really appears to be a technology for the future. Again, with DNA vaccine, as you know, you can send the DNA, which will come as a vaccine. Cells take it up, transcribe into RNA, and the RNA makes protein, and for the spike protein. The RNA, you're cutting one step, make the RNA in vitro, package it in a lipid particle, and give that as a vaccine. RNA goes into cells and translates into protein. The other approach, again, viral vector vaccines largely uses adenoviruses, like the Oxford vaccine uses a chimp adenovirus, the Russian vaccine uses human adenovirus, and so on. But again, the idea is to take the gene for the spike protein and try and make the spike protein in cells. And finally, the virus vaccine, the protein vaccines, the virus-like particle is the new Novavax vaccine that is coming up and has shown quite good results in trials. So this is a summary of vaccines that are available at this time. Here is the pipeline. 4 vaccines have been approved, 6 have been authorized for limited use, and roughly 60, 65 other vaccines are in human clinical trials. This is a list of the leading vaccines. The 2 that are available in India right now are the Oxford AstraZeneca vaccine, which is made by the Serum Institute of India. And the other, so the Oxford AstraZeneca vaccine is a chimp adenovirus-based vaccine. The other vaccine available to us now is the Bharat Biotech vaccine by the name of Covaxin, which is an inactivated viral vaccine. And both of them so far have emergency use approval in India. And the plan in India is to vaccinate 10 million healthcare workers, 20 million frontline workers, and then 26 million people, 260 million people who are above the age of 50, and another 10 million who are below 50 but have significant comorbidities. So that's the plan that is rolling out in India. So in the end, would current vaccines work against variant viruses? And so there's been a number of publications on this, and I'm not going to go through each of them. And as you see, most of them have come out in the last 6 weeks or so. Most of them are preprints. Let me simply summarize using this paragraph from NEJM article that was published last week, which essentially says that the full set of B1351 spike mutations, which is essentially the South Africa lineage virus, is not neutralized very effectively by vaccine-derived antibodies. And the 2 critical mutations in that are the N501Y and the E484K mutation. And both of these mutations are at the interface of the receptor binding domain binding to the antibody, shown in more detail here. So if I can take you back to your undergraduate biochemistry and come back to the table that I showed you earlier about mutations in Indian isolates, these are the mutations that have so far been found in India. And as you will see, this is really the most drastic of those changes, changing a negatively charged amino acid into a positively charged amino acid at the binding interface. And this mutation as well, which brings in an aromatic amino acid in that location. The 4 mutants that have been seen in Maharashtra, this change is not as drastic a change going from glutamic acid to glutamine. Again, showing you the mutations that have been mapped in India so far. And as a summary, there are some mutations of concern that are emerging in India. But so far, these are single mutations, and they have not come together in any organized lineage in the country. The B117 or the UK variant is present, but so far, no community transmission of this has been reported. But that doesn't mean it won't happen in the coming times. Therefore, genomic surveillance is really needed, and it's needed at a reasonable density. And that's where INSACOG comes in, as Soumitra told you earlier. And this is something on which your own institute is leading the national effort of 10 different laboratories. The idea is to sequence at 5% density of confirmed cases. And hopefully, if we are able to do that successfully, then we will catch any emerging mutants and any emerging lineages well in time to be able to put public health measures in place. So with that, I will stop. And thank you very much for asking me to give this lecture. Thank you.
Thank you, Professor Jamil, for this illuminating talk. Next, we have the keynote address, which will be delivered by today's guest of honor, Professor Sarah Teichmann. Parthapratim Majumdar to kindly introduce Professor Teichmann.
Hi, Sarah. How are you? Are you able to hear me?
I'm very well, thanks. How are you? Can you hear me?
Yes, we can hear you. Thank you very much. First of all, our deepest gratitude to you for accepting our invitation to be the guest of honor on our Institute Day and to deliver this talk, which we eagerly look forward to. Let me introduce you to the audience, and I'll be short. I won't embarrass you too much. Dr. Sarah Teichmann is currently, since October of 2015, been the Director of Research at the Cavendish Laboratory in Physics Department of Cambridge University. Since February of 2016 and October of 2016, she's been Head of Cellular Genetics at Wellcome Sanger Institute and concurrently a Senior Research Fellow at the Churchill College. Her background is in biochemistry, and of course, she performed very, very well in her coursework, both in the undergraduate and graduate studies. She, in spite of having a biochemistry background, she predominantly does computational biology and bioinformatics. She got her PhD in those areas, combining sort of interdisciplinary, but she mostly focuses on computational biology and bioinformatics. What does she do? She asks big questions on immunology. Currently, she's trying to understand the dynamics of immune response, and she uses interdisciplinary approaches, primarily bioinformatics and functional genomics, such as single-cell RNA sequencing. And we are poised to hear how to reconstruct a human body cell by cell. So we'll hear a lot of single-cell RNA sequencing. Earlier, she did structural bioinformatics, but now she spends most of her time doing single-cell RNA sequencing. She did structural bioinformatics, exploring the global principles that drive the assembly of protein complexes. She's moved on, and she's trying to reconstruct the human body cell by cell. We consider her to be a leader, and she leads the Human Cell Atlas project, with which I'm associated. She and Aviv Regev, who was up until recently with the Broad Institute, but has now moved on to Genentech, she and Aviv, they co-lead the Human Cell Atlas project, which again is understanding every human organ cell by cell and informing human disease. Sarah is a fellow of the Royal Society. Last year, she was elected a fellow of the Royal Society. She is, in 2015, she was elected a fellow of the Academy of Medical Sciences. She's a fellow of the International Society for Computational Biology. She's an EMBO Gold medalist. She's received the Colworth Medal of Biochemical Society, Helmholtz International Fellow Prize, Genetic Society Mary Lawrence Prize, Biochemical Society GlaxoSmithKline Award, and I'm sure that I have not exhausted all of the various awards and medals that she has received. It's really our pleasure to welcome Sarah Teichmann, and we look forward to your talk, Sarah. And it's really been my personal pleasure to be associated with her and work under her leadership. We hope that many of us in the Institute will undertake single-cell sequencing work, and we will get into closer contact with her and her entire lab in the Wellcome Sanger Institute. Sarah.
Thank you so much, Partha, for that kind and very generous introduction. And thank you so much for having me here today. It's a real pleasure and an honor. I'm gonna try to do share screen now.
Yes.
Can you see this? Can you see this, Partha?
Yes, absolutely.
Fantastic. Thank you so much, and thank you for introducing the Human Cell Atlas, and also a big thank you to Partha for being a partner on this Human Cell Atlas journey and for being part of the leadership of the consortium from the very beginning. So what I'm going to talk about is mapping the human body one cell at a time. As Partha said, that's really the work that I'm focused on at the moment. And of course, we all know that each tissue in our body consists of a collective of cells that are working together in the tissue to deliver and create the function of the tissues and ultimately the organs in our body. And it's the collective of genes that are active, that are switched on in every single cell that lead to the different morphologies and the different functions that the individual cell types have. So the neuron has a very different function from the fibroblast, yet of course the DNA inside each cell is identical. But it's the invisible machinery that regulates the ensemble of genes that are active in the cell that then makes the different set of proteins in each cell that leads to the different morphologies and functions. And as Partha said, it's the single-cell genomics technology that's a really powerful way of profiling and understanding the RNA that's active in every single cell in a comprehensive and unbiased way. And in that sense, the resolution revolution in genomics that allows us to isolate individual cells and sequence the nucleic acid content of each individual cell is kind of better than a microscope in terms of dissecting the phenotype of each cell. And this technology, this resolution revolution in genomics that allows us to resolve the nucleic acid content of individual cells in a high throughput way, it was one of the technology innovations that really catalyzed the Human Cell Atlas initiative. And what this project is basically an international consortium aimed at creating a comprehensive reference map of the types and properties of all cells as a basis for understanding, diagnosing, monitoring, treating health and disease. And it's what we want to do together is to map the cells in our body, not every single one of the 37 trillion cells, but all capturing all the cell types and cell states. And as Partha said, this goes back to actually 2015 or early 2016, when Aviv and I started talking to each other about how to organize an international project like this. And what we set out to do was to gather the community to create this Google map of the human body. And it's now got over 2,000 members from 77 countries, almost 1,200 institutes, only 45 members from India. So please sign up here. It's an open consortium, a grassroots initiative that's open to all scientists. You can simply click on humancellus.org/join HCA and fill out an easy form, and then get the newsletters and updates, meeting information. We have lots of online seminars now in different time zones to share basically the research that's going on in the consortium. Parth, as I mentioned, is part of the original organizing committee, the governing body of the consortium, and he's also done tremendous work on equity, diversity, global outreach, together with Alex Shelik and Musa Mwanga, to increase the reach of the project to all areas of the world. The other organizational component is the biological networks, where we are organized also into the different tissues and organs of the body, and there are leaders for each of the individual systems. So you've got the eye, the nervous system, heart, lung, pancreas, kidney, reproductive tissues, human development, organoids, so also using the human cell atlas to compare to in vitro systems, and then genetic diversity as well, linking also to equity and diversity. So as I mentioned before, it was really the transition from conventional bulk genomics, which is sort of the milkshake or fruit smoothie kind of a view of cells that are hundreds or thousands of cells that are averaged, to the single cell genomics technology that was one of the technology drivers of the formation of the Human Cell Atlas Consortium. And then hand in hand with the single cell genomics, it's also the spatial methods, spatial transcriptomics and other spatial mapping methods that are telling us how the cells work together in their three-dimensional organization in tissues. And of course, all of these experimental methods, the single cell genomics plus the spatial transcriptomics, are tied together with computational processing, analysis, integration techniques, as Partha mentioned. And it's really the work across these different disciplines that allows us to go from the organ picture all the way down to the single cell to define the individual cells and tissues. And so what I want to focus on in the next half an hour or so is what's the impact of the Human Cell Atlas project in terms of how does understanding the cells in our tissues help us in a practical sense. And there's one context which is regenerative biology, regenerative medicine, and there's another context which is disease mechanisms. I'm going to talk about both of those to show you how a basic profiling of and a thorough, comprehensive, deep understanding of the healthy reference cells can help us in these practical applications to regenerative medicine and disease. So if we look at the amount of data that's been generated by that huge global community to date, then you can see here a few snapshots from 6 different tissues. I'm showing the kidney on the top right here, where we've got almost 500,000 cells, and the little virus beneath the tissue is COVID-infected cells. So there are several centers, including ours, who've been sequencing post-mortem and analyzing post-mortem tissues from deceased COVID-19 patients. For the skin, there's 1.5 million cells. The airways, almost 3.5 million. The gastrointestinal, the colon here, or the gastrointestinal system, almost 2 million from hundreds of individuals. Human development, which is obviously a conglomeration of many of all the different tissues, is almost 6 million. And the liver, almost 500,000. So you see that for at least for these major organs in the adult and also for human development, we're really starting to make very good progress on a Human Cell Atlas to get to version 1.0 of the Human Cell Atlas. I'm going to give you a snapshot kind of a couple of different pieces of work and different projects of the that are going on that my lab is involved in. And in particular, I want to tell you about our Human Cell Atlas of the uterus, and that's now on bioRxiv and it's under review. And this was started off by Rosa Vento-Tormar as a postdoc in my group, and she's really taken on the project now in her own independent group, which is a rising, growing group within my program at the Sanger Institute. So the uterus is a really fascinating tissue that is quite human specific, And there are even elements of it that are specific to humans and that we don't share with other non-human primates. And they include things like the spiral arteries, so the shape of the blood vessels in the uterus. And of course, the endometrial shedding, the menstruation, is specific to humans and is not found in rodents. And it's really that rapid evolution of reproductive biology, which is known basically from genome sequencing. And it's also the dynamic nature of this tissue on the monthly cycle that means that it's relatively poorly understood as compared to, let's say, the airways or other tissues. And so we set out to create a cell atlas of the endometrium throughout the menstrual cycle to cover these different stages and the different tissue architectures that are found within this dynamic organ. And so, as you know, there's a sort of there's a control of or a regulation of the tissue by hormones, estrogen and progesterone, that lead to a window of ovulation and then a window of implantation before you have finally the shedding and the restart of the monthly cycle. And so the different tissue samples that we got was from organ donors, so deceased organ donors, women who die at different stages of the menstrual cycle and agree to provide their organs for transplantation and research. And we work very closely with the Cambridge Biorepository for Translational Medicine, which are our colleagues in transplant surgery in Addenbrooke's Hospital here in Cambridge. We also analyzed biopsies, which you can see are kind of just the upper layer. So these are from living patients that are having biopsies for diagnostic purposes. And those are only a couple of millimeters thick sometimes from the top of the tissue, whereas the organ donor biopsy goes through the full thickness, through the entire endometrium and into the myometrium, which is the muscular outer layer of the uterus. And so the deceased transplant donor biopsies are very precious in that sense because they give us this full thickness biopsy of the tissue, but they're quite rare because they're over a year. There are not many women, female donors that are premenopausal, thankfully so, but it means that the tissue is very precious. And so what we have is 4 transplant donor samples from the follicular phase and 3 from the luteal phase. And we now, this is outdated, we now have some additional donors, but the number of end donors is limited for the 2 different phases that we have. What we've done is to combine single nuclear RNA sequencing, so where we extract the nucleus from fresh or frozen tissue and profile the transcriptomes, with Visium spatial transcriptomics, where we put a tissue section onto these 10x genomics chips that are 6.5 by 6.5 millimeters. And each voxel here, each little circular feature is 50 microns in diameter, and there's 50 microns in between, 100 microns from the center of each feature. And in that way, we get not only from the nuclear data the information about the cell states in the tissue, but we get also the kind of transcriptomic landscape of the tissue section in 2 dimensions. And so here you can see a tissue section from the luteal phase, the second half of the menstrual cycle, where we can score basically the glandular transcriptomic signature on this tissue section and find regions that correspond to glandular epithelial cell signatures. We can define different fibroblast or stromal cell zones in the tissue. So you can see here the decidualized fibroblast that we already described in our Nature paper in 2018 on the maternal-fetal interface, the placenta-decidua interface. You can see that fibroblast signatures at the top of at the inside towards the lumen of the endometrium, and the basal fibroblasts are at the interface just above the myometrium, at the interface between the myometrium and the endometrium. So in this way, we can define the different zones of the endometrium. It's a kind of layered tissue, sort of like your skin, but it's defined by different fibroblasts rather than different keratinocyte states. From the bottom to the middle to the top, I'm not going into all the fibroblast cell states, just the basal and the decidualized fibroblast cells here. If we look at the epithelial cell states, I've mentioned the glandular signature here. We can also see ciliated epithelial cells on the luminal on the inside, towards the center of the uterus at the top of the endometrium here. And we can discover for the first time a new progenitor cell state for the endometrial epithelial cells that's marked by LGR5, which is also located here, interestingly, in the luminal side in the luteal phase, in the second half of the menstrual cycle. These progenitors are actually interestingly located inside the cell. And understanding these progenitor cells is important for interpreting endometriosis and endometrial cancer cell states and relating to what the pathological cells are in these diseases, in these very important diseases. Now, I've mentioned spatial transcriptomics as a sort of general category of transcriptomics, and I've talked to you about how valuable and powerful the sequencing of tissue sections can be for spatial transcriptomics. What you see here is a different flavor of spatial transcriptomics, and this is multiplex single molecule FISH, where we have 4 different colors marking DAPI and then 3 genes, EpCAM, matrix metalloprotease, and PAEP, for zooming in and getting a very high resolution view. So each square is amplified here in below, so that you can see the even subcellular resolution of distribution of gene expression. And so these imaging methods can drill in and give us a higher resolution picture of a tissue, and are very complementary to the sequencing-based methods that I showed you before, that are kind of giving us a more coarse-grained view of the tissue architecture in 2 dimensions. And so what this is basically, in this slide, I'm not showing you any new cell types. It's just basically to understand how the tissue changes in morphology and shape from the proliferative phase through mid to late proliferative, secretory, mid-secretory, and late secretory, to kind of get a dynamic time course of how the glands that you can see here are forming through the proliferative phase. You can see they're very plump and puffy here. Then the secretory phase, they kind of fuse and then disintegrate finally before the shedding occurs. And so the value of the imaging in this particular image is less discovery, but more in terms of morphology and high resolution structure of the tissue. So this is marking the epithelial cell types. What I showed you before was focusing on the fibroblast and the epithelial localization at a more coarse-grained way. Okay, so in order to use the information from the tissue to understand how to engineer cells in vitro, we want to be able to know what the transcription factors are driving or controlling or regulating each cell state. And what we used for this was Dorothea to identify the transcription factors in the ciliated cells and the secretory glandular cells. And so what you can see here in the rows are the different epithelial cell states that we're describing: pre-ciliated, ciliated glandular, a glandular secretory cell, and then 2 different luminal progenitors that I mentioned. And if just mapping these cells onto the 2-dimensional tissue architecture, you can see here the different layers where those luminal progenitors are located there with the ciliated cells. Then we've got sort of functional secreting glandular epithelial cells in the middle, and then the basal layer at the bottom. And at the top, you can see the transcription factors that are controlling ciliated differentiation. So pre-ciliated, ciliated, FOXJ1 has 6, and so on. At the bottom, the other lineage, which are the glandular epithelial cells, where you've got FOXO1, HEY1 has 4, and so on. So these are other transcription factors that are controlling that lineage. And so we use bioinformatics to deconvolute the single-cell genomics data and find the key transcription factors regulating each one. Now, the transcription factors themselves are responding to signaling pathways, and we can find transcription factors that are responding to Wnt activation, Wnt inhibition, and Notch activation here in these 3 different colors. And so you can see that the ciliated lineage is controlled by Wnt activation in turquoise, whereas glandular is more Wnt inhibition and Notch activation. And this is by integrating previous data. Now, where are these Wnt and Notch signals coming from? In order to look into the tissue microenvironments and deconvolute the signaling pathways, the signals that are coming into the epithelial cells from the transcription factors, we unravel the signals that are coming from fibroblasts to epithelial cells using our database cellphonedb.org. And we developed a new version here where we integrate the single cell RNA sequencing or single nuclear RNA sequencing data with spatial transcriptomics and information about the receptor ligand complexes, taking into account their stoichiometry. So Partha mentioned that I'm a protein complex geek, and I like to be very specific about the correct stoichiometry of receptors that's required to make a functional complex. And by integrating that information with the single cell RNA-seq and the spatial transcriptomics, we can predict high confidence cell interactions. And so this gives us Wnt activation for the ciliated cells and Wnt inhibition and Notch activation for the secretory cells. Sorry, this is a bit misleading. This should be shifted upwards on the slide. In any case, what we can use is now leverage those signaling pathways, those signals and the information about the transcription factors, and connect this in vivo HCA information with the ambition of designing or creating cells in the dish, creating in vitro models in the dish. And so here we're using, in this particular case, we're using endometrial organoids. And so we're effectively taking the recipe for making the ciliated cells and the secretory cells from that signaling pathways and the transcription factor pathways that we learn from the in vivo human cell atlas to engineer an endometrium in a dish. And so we take the tissue sample from the endometrial biopsy, digest, have cell fragments in matrigel and establish the cultures. And then the way we engineer this system is to add Wnt and Notch inhibitors. And these are small molecules, they're chemicals that we add into the culture. And we can then characterize those new organoid by both microscopy and by single cell genomics. And that we've done that's described in this paper online. I'm not going to go into the details. And basically engineer the organoids to be either more ciliated, like these cells on the lumen that are activated and go down this differentiation pathway in response to Notch signaling, or the glandular cells that are sitting more deep inside and doing the secretions, and they're activated by Notch signaling. And of course, it's the Wnt signaling that also has impact on endometriosis and endometrial cancer, as is known, and we can now kind of relate those signaling pathways to the hormonal responses. And so I hope I've convinced you that basically the atlasing of tissues in vivo is informative for making models in vitro, which ultimately have relevance for things like transplantation, but also for drug screening, in addition, of course, to a more deep mechanistic understanding of the cell biology of human tissues. What I want to go on to now is the other impact of the human cells, which is disease mechanisms. And basically start off with a story about the human airways, where over 5 years ago now, we started a project in collaboration with GlaxoSmithKline and Open Targets, where we were studying both the human bronchi and the human parenchyma, the alveoli. So the large airways of the human lung, as well as the lung parenchyma, with the aim of understanding the difference between healthy and asthmatic lungs. And this was driven by GSK's, or funded by GSK's, interest in asthma, which is, of course, a thickening and inflammation of the airways that leads to a tightening and attacks. And there are some steroid responsive asthma, but some asthma that's non-steroid responsive. And so there's still a need for development of drugs in that area. So what we did here was we teamed up with Martijn Nawijn and colleagues in the Netherlands in Groningen to study biopsy tissue from little pinch biopsies from bronchoscopies from healthy volunteers and from asthmatic donors that are taken from between the 3rd and the 6th generation of the airways basically where they divide. And then getting lung parenchymal tissue to profile the alveoli from our deceased transplant donor program here in Cambridge with Cora Saib Parsi and colleagues in the Cambridge Biorepository for Translational Medicine. And we studied these cells both by droplet single-cell transcriptomics, but also by plate-based protocol that we set up robotically in my lab. And we published this work a few years ago. And one of the key things that I want to highlight here is also the different goblet cell populations that we found in nasal biopsies. So in addition to the 2 locations of the lung, or in addition to the bronchial biopsies, the Dutch group also profiled some cells from the nose. And so we found not only different cell types in the lower airways, but also these goblet cells and on top of the ciliated cell types that are specific to the upper airways. And in particular, we found one goblet cell population that we described as being more immune signaling, because these cells were secreting signals that had the complementary receptors on T cells and dendritic cells. And so we defined these as being a kind of immune signaling goblet cell type on top of the less immune activated goblet cell type. Now, the year after we published this work, of course, the SARS-CoV-2 pandemic emerged, and Shahid has just talked to you about that and the different strains. And I'm going to continue that story because I guess has been on all of our minds as such a huge public health emergency across the entire world over the last year. And I'm sitting at home because we're still in lockdown number 3 here in the UK. And what you can see on the top of these cells is the ACE2 receptor, which is the receptor, the enzyme that's the receptor for the SARS-CoV-2 virus. And of course, Shahid has just taken you through all of this cell biology and molecular biology, where the coronavirus sits docks onto the cells that have the ACE2 expression and is then internalized via cleavage with proteases, TMPRSS2, and so on. And so what I realized in discussions with Human Cell Atlas colleagues, in discussions with Jeremy Farrar, whom Shahid knows is the director of Wellcome, and we've been on a course together funded by Wellcome. And I had a discussion with Jeremy and mentioned that we'd found these cells inside the nose that have the high expression of the viral entry receptor. And together with the Human Cell Atlas community, as our first consortium paper, we then mapped the entire Human Cell Atlas data with respect to the presence of SARS-CoV-2 entry receptors. And I have to say that this was really an incredibly exciting project, because within a couple of weeks, it wasn't only the published data, but people were sending us their counts for the viral entry receptors, even in unpublished data like the cornea from Linda Lako, the gallbladder from Ludovic Vallier's group, heart data, and so on, so that we are able to make a very comprehensive map of ACE2 across healthy human tissues. And I'll take you through just a few of the barrier tissues, the barrier tissues to the outside world. So I've mentioned the nose, where we found very high expression of ACE2 and TMPRSS2, which is marked in dark blue in these goblet cell types, but also ciliated cells. In the lower airways, in the bronchi, club and ciliated cells have expression. And then, of course, it's famously and known for a long time for SARS-CoV-1 already that alveolar type 2 cells in the parenchyma have high expression of the viral entry receptors. And then if we look in the eye, I mentioned that exciting data from Linda Lako's group on the eye. We find that there are superficial conjunctival cells that have high expression of the viral entry receptors, and this can explain potentially the viral conjunctivitis that's seen in SARS-CoV-2, and also implicate the nasolacrimal tract in transmission from one tissue to another inside the body. In the gastrointestinal tract, it's known that there are enterocytes that have high expression of ACE2 and TMPRSS2 and the other proteases. And at the maternal fetal interface, so the endometrium I talked to you about before, there are perivascular cells on the maternal side and on the fetal side in the placenta. when there's implantation, there are syncytia trophoblasts that have high expression of the viral entry receptor. So this could provide a potential mechanistic explanation for vertical transmission that's been reported rarely, but there have been cases where vertical transmission has been reported. And so this was a very exciting project that Wardon Songnak, an incredibly talented Thai postdoc in my group, and Ni Huang, a staff scientist working with me, coordinated with input from the entire human cells community, in particular the lung biological network, where this sort of started off, Pascal Barbry, Martin Navain. And then I mentioned some of the labs that were generously prepared to share the cell by gene counts for the viral entry receptors of unpublished data from the eye, the heart, the gallbladder, and so on. And we made this data available at covid19cellatlas.org. It came out last April in Nature Medicine. It's been cited going on for 1,000 times, and the data is available at this website and has been accessed tens of thousands of times. Now, the next step going on from that kind of predictive survey is obviously to analyze patient samples and to look at both blood or respiratory samples or the autopsy tissue from patients. And so I'm going to go on next to talk first about some of the respiratory and oral samples that we've been studying, and then the blood quickly at the end. And so we expanded the covid19cellatlas.org site to include these patient donor samples from the blood and the tissues. There's a short paper on medRxiv kind of talking about this resource in the context of all the single-cell genomics blood projects around the world that have been supported by the Trans Zuckerberg Initiative. So we ingest and serve that data. Now, in unpublished work, what we did with Marko Nikolic's group at UCL, and Carsten Meyer from my group, and again, Ni Huang and Christoph Polanski were analyzing the data, and we're studying nasal scrapings of COVID-19 patients. And I told you before that we predict these goblet cells, these secretory cells, to be viral entry points inside the nose, which can explain some of the high transmissibility of this virus. And indeed, when we look at nasal scraping data, we can find reads that are mapping to the viral genome. So the blue is the human genome mapping RNA molecules, the orange are the viral mapping molecules, and indeed they're highest in these goblet cell, secretory cell populations. And interestingly, we see this inflammatory epithelial cell state where there's a massive antiviral innate immune response in the epithelial cells in infected individuals. There's also some viral reads in ciliated cells, as expected, as they also have the receptors, albeit at lower levels. Now, in the oral cavity has kind of been late to be mapped by the human cells community, but Blake Warner from NIH and Kevin Bird from University of North Carolina teamed up with us. Again, Ni Huang in my group analyzed the data to study biopsies from different parts of the oral cavity, both in mouse and in human. The human is here. And what we can see is that indeed in the mouth, we have TMPRSS2 ACE2, the viral entry receptors, in ductal epithelial cells in salivary glands. And so what this suggests is that the virus could enter into these higher up epithelial cells in the salivary glands. And if we look into imaging data from COVID-19 patient biopsies from the salivary glands, we do indeed see the viral RNA inside these cells. And this is on medRxiv, and it's coming out in Nature Medicine soon. So again, the viral entry pattern in the healthy reference human cell atlas data predicts where the virus is going to be sitting precisely inside the tissue. And so finally, in the last part of this talk, I'm going to go on to analysis of blood data from different cohorts of COVID-19 patients, mild, moderate, severe, critical, that I've been involved in with my lab in collaboration with many teams here to integrate data from across the UK. And so what we did here, like many of the other groups around the world, was leverage the human cell atlas infrastructure in terms of genomics, bioinformatics, sample processing, and work together as a team science, in a big team science way across many teams, to use single-cell multiomics, site-seq, protein and RNA combined data with TCR and BCR sequencing to study samples from 3 centers in the UK, Newcastle, Cambridge, and London. And so this is a big collaboration across many groups from Newcastle, London, Cambridge, Sanger, shown here. And we all work together to really, in a record speed and a lot of time pressure, to put this data together. It's on medRxiv and it's under revision now, so you can read about this in more detail. The cohorts include asymptomatic COVID-19 infected individuals, which are super interesting because they're quite hard to study. Not many papers have asymptomatic cohorts, and then mild, moderate, severe, and critical cohorts, as well as controls, healthy controls, hospitalized non-COVID controls, and IV LPS controls. And so the total cohort is about 130 patients. We looked at site-seq surface proteins, as well as 5' gene expression, TCR and BCR patterns. And what this gives us is a very detailed, it's actually 1.5 million, not just 850,000 cells, a very detailed map of the different cell types in the blood, including T cells, innate lymphocytes, the myeloid compartment, the B cells, and also progenitors. It's a very, very detailed map of these different cell states. I think more than has been available before. So it's interesting even understanding our blood in a lot of detail. What we discover is basically cycling myeloid cells, cycling monocytes that other people have also described as being implicated in the disease. In the asymptomatics, we see megakaryocyte priming from progenitor cells, which is interesting in the context of the coagulopathies. We see a difference in the T cell compartment between asymptomatic and severe patients in the sense that asymptomatic donors have a high T follicular helper cell abundance, relatively speaking, and a correlation of the T follicular helper cells with plasma cells in the blood. They have a slight but not highly significant enrichment in regulatory T cells, and they have a high IL-22 CD4 T cell substate. So we think that the asymptomatics really represent a healthy immune response where you have a balance between T and B cell response, but also some regulatory cell upregulation to avoid the overactive immunity that we see in the severe patients where you've got clonally expanded CD8 T effector memory ratio, proliferating CD4 and CD8 cells, high plasma cell, high IgG, high inhibitory BCR signaling. And that's really the pathological response that's then overactive and leading to a lot of the complications of the disease. So it's not the virus itself, but it's actually this overactive immune response that's leading to the severe pathology. In the asymptomatics in the B cells, there's more IgA than IgG and increased TNF-NF-kappa B signaling. And the mild and moderate, we see greater clonal expansion of B cells in females than males. So that's really the B cell compartment. And so in summary, I've told you also about how the Human Cell Atlas Healthy Reference, but also the technologies and the team science framework has given us insight into disease mechanisms in the context of asthma, but more COVID-19 that I've discussed in terms of both the viral entry receptor pattern, but also the immune response. And so I hope that through this COVID-19 story of the Human Cell Atlas community and the regenerative biology organoid, endometrial organoid endometrium story, I've convinced you that the Human Cell Atlas project is valuable not just as a basic science project in terms of understanding the cells in our body, but also informing us how to what's happening during disease and how to combat it. With that, basically, I would like to wrap up this story about the uterus, which of course is key for development, and the airways and other tissues implicated in SARS-CoV-2. Thank you very much for listening. Thank you for this opportunity to present here, and I'm happy to take questions.
Thank you, Professor Deichmann, for an enlightening talk. Before we end today's proceedings, I would request Dr. Bhashwati Pandit to kindly deliver the vote of thanks. And I do not think we have wherein we can take questions today.
Okay, that's fine.
Yeah. Sorry about that.
No, not at all. Thank you for having me.
Hello, can you hear me?
Yes, yes, you can hear you.
It's an honor to have been asked to offer the vote of thanks on the occasion of the Institute Day of the National Institute of Biomedical Genomics. On behalf of NIBMG, I'm extremely thankful and extend my gratitude to our honorable and respected chief guest, Professor Shahid Jamil, and guest of honor, Dr. Sarah Teichmann, for accepting our invitation and sharing their illuminating ideas and the knowledge showered on us. We have been fortunate to have you both amongst us in this auspicious event, and all the participants of this meeting are inspired by your highly motivating and sparkling scientific discoveries. I thank our director, Professor Soumitra Das, for his leading supervision and inspiration at every point of time in organizing this event. We are also delighted to hear the genesis of the National Institute of Biomedical Genomics from Professor Parthapratim Majumdar, without whose hard toil we would not have been standing here. This is particularly inspiring to the youngsters, the students who have recently joined the institute and not aware of what NIBMG was 10 years back. I'm also thankful to all the distinguished guests, participants, and audience for attending this event and sharing with us some of the finest moments. I take this occurrence to thank the entire organizing committee, all the subcommittees, all faculty members, managers, all administrative staffs, students, and all the members of NIBMG for sharing the load and execution of all the events smoothly. I would like to also thank all of you who has helped us directly or indirectly to make this celebration successful. And this brings us to the end of the scientific session. And once again, I thank you all.
Now we break for half an hour for high tea. Thank you.
Thank you, Sarah.
Thank you. Bye.
Really, really
Enjoy your
enlightening talk.
Bye.
I hope I can join in person another time.
Right. We are going to miss you here. I think next time you come and visit us, both Shahid and Sarah.
Hi, guys. Thanks very much. Soumitra, you joining us at 5?
Yes, I will
Okay.
wait for that.
All right. See you at
Sure.
5 then.
Yeah.